Hi dear students, welcome back to one more session of bioinformatics, and today we are going to talk about two softwares which are mainly being used for the analysis of DNA as well as the protein sequences in the biological databases. And the most commonly used softwares of these are the BLAST as well as the FASTA. Now do not get confused what is FASTA. FASTA, we had mentioned about the FASTA format of the DNA sequence earlier. But at the same time, there are two softwares, as we mentioned here, BLAST as well as the FASTA. These are mainly softwares which are used to align a query sequence to various sequences which have been provided in the database. So let's just explore them. What are these sequences? What are these softwares? And basically, these are considered to be as two major heuristic algorithms. Now, what do you mean by heuristic algorithms? Okay, that is with our own, it's a software or an algorithm which can be learned by ourselves itself, and a person can learn something for themselves itself with a hands-on or interactive approach of learning. And among this, you can see that the BLAST software is mainly been provided by the NCBI database, and the FASTA software is mainly being provided by the EMBL database. So when we come across large amount of DNA as well as protein sequences in the public databases, and we have one particular sequence of our own interest, probably we would have to find out what is the similarity of our sequence with the sequences which have been provided in the database. So to analyze them, we use these softwares. You need some algorithm based softwares to carry out this, and FASTA and BLAST are most frequently exposed or frequently used softwares which we come across when we need to find out a similarity search of either a nucleic acid sequence or a protein sequences with a biological database. So at the beginning, now we are going to explore the BLAST software. So let's go explore it. So now let's work with FASTA as well as BLAST. FASTA and BLAST, as we know, they are softwares which are used in bioinformatics, and both of them use a pairwise alignment of the nucleic acids as well as the amino acid sequences based upon what are you searching. And it has been found that short stretches of identical or nearly identical letters in two sequences are being found out when you do a BLAST or a FASTA sequence. And these short strings of characters are usually considered to be as words. The basic assumption is that two related sequences must always have at least one word in a common. And by identifying the word matches, a longer alignment can be obtained by extending the similarity regions from the words. And once the regions of high sequence similarity is been found, the adjacent high scoring regions can be combined into a full alignment. But you should remember that both of these FASTA and BLAST, they are taking some sequences and they are aligning them with identical letters or nearly identical sequences which are being found in the biological database. In the case of BLAST and FASTA, but though they use almost the same pairwise alignment technique that they use here, you can find that BLAST is mostly involving in finding of ungapped locally optimal sequence alignments, whereas FASTA is mainly involved in finding similarities between less similar sequences. Now previously in the pairwise alignment, you can just have a look at this particular slide. You can see that when you align two sequences here, the gapped locations are coming here. That is there are spaces in which when pairwise alignments, when there was a dissimilarity, a gapped sequence was being found in this pairwise alignment. Now in the case of BLAST, such an occurrence of gapped regions normally do not occur in the case of BLAST. Ungapped findings are being obtained in the case of BLAST. And often we require to get similar sequences, or the pairwise alignment is following that similar sequences or identical sequences get aligned in the case of BLAST. If it is not identical or if it is not similar, we will say that the percentage it has no much similarity, such a result will be obtained. But in the case of FASTA, they find similarities between less similar sequences. You need not have to have much of similarity between the sequences when you are using a FASTA software. Now let's see what is BLAST. BLAST is expansion of the basic local, it's a short form of basic local alignment search tool, and it was a program which was developed by Stephen Alsole of NCBI in 1990 and has since become one of the most popular programs for the sequence analysis. Here it uses a heuristics to align a query sequence with all the sequences in the database, and the objective is to find the high scoring ungapped segments among the related sequences. And the existence of such segments above a given threshold indicates the pairwise similarity beyond random chance, which helps to discriminate related sequences from the unrelated sequences in a database. And it is a popular bioinformatics tool due to its ability to identify regions of local similarity between two sequences very quickly. And here it will also calculate an expectation value which estimates the number of matches between the two sequences, and it uses a local alignment of sequences. So basically what you do, they try to find out ungapped segments which are having among the related sequences, and how much percentage of similarity do you get, as well as what is the expectation value of that, and that is the number of matches which has been possible between them. Okay, and this BLAST can be accessed in different databases right now. It was firstly started by NCBI database, but you can also see that in different databases also this the BLAST software has been used. Now in Canadian Bioinformatics Resource BLAST you are having, European Bioinformatics Institute also you have BLAST, like various other websites are also providing access to the BLAST software right now. Now this is the NCBI BLAST page which is being shown over here, and you can see there is a BLAST. If you go to NCBI, you have a BLAST thing. If you want to blast the nucleotides, you have the BLAST, and you will have N BLAST, or if you have the protein to be BLAST, you have the P BLAST or the protein BLAST. Then this is the BLAST server. The BLAST N is being provided here, showing that what you want to. If it is a nucleotide, it is a BLAST N, and if it's a protein, as we told, it's a BLAST P. And here you can either upload your DNA in the you can upload a file from here which has been provided in the FASTA format, or you can just paste the sequence over here and give a submit button. Now this in EMBL's database also you can find the BLAST sequence here. And here what does it means? Now I am taking a protein BLAST over here in the, so that's what I need to tell you, that in different databases you can come across the BLAST sequence. It's not that now it is only there in the NCBI database, it has been found in various other databases. You can find the link to the BLAST sequence, and there are different flavors of BLAST. Now if you want to if you want to blast a protein, you have the BLAST P. If the nucleotide, it is a BLAST N. It can, now this BLAST N can be either DNA or RNA. Query against the GenBank. Then you have BLAST X, that is 6 frame transitions of the DNA query sequence against a protein database, that is possible. Then the TB BLAST N, where a protein query is being blasted against 6 frames of the GB translated, and then GenBank translate. And then you also have T BLAST X, where 6 frames of the DNA query sequence is blasted to 6 frames of the GenBank translate. And the Psi BLAST, where the protein profile query against the protein database. Then the Phi BLAST, where the protein pattern against the protein database, that is also there. So these are the different flavors of BLAST. That is the same basic local alignment search tool. It's a basic local alignment search tool, but what is being blasted over here is being found to be different. And other BLAST services, you have Mega BLAST, you have RPS BLAST, BLAST 2 sequences, genomic BLAST, as well as VEX screen and all. In Mega BLAST, you compare large sets of long DNA sequences. In RPS BLAST, you conserve domain detections are being possible. In BLAST 2 sequences, you take pairwise alignment of 2 chosen sequences. Then genomic BLAST, you align against selected human, microbial or malarial genomes. That is, you will be having sometimes, now we had come across a Saccharomyces cerevisiae database. So sometimes you might take a DNA sequence and you might align it against the Saccharomyces cerevisiae database. So that is a genomic BLAST. That is, you are taking the genome of Saccharomyces and your sequence is being blasted against that. Then you have the VEX screen for determining the cloning vector contamination in a sequence data. That is, when you sequence the data after recombinant DNA technology, sometimes some of the sequences of the vector like PET32 and all that, there are different vectors, okay, which can come across. So that vector, you need to find out in the sequence data if any vector or cloning vector's contamination has been found. For that you will use the VEX screen. Then you have, this is how to run it. We will be seeing a recording of that, how it goes. You just have to place it over here and put a BLAST. You will get a sequence, a result. And this is a protein sequence. Now if you want to blast of a protein, you can take this amino acid sequence and put it for blasting. And you can also, this is also in which you can paste the sequence, the FASTA format you are putting. Either you can put the accession number or you can just space this particular, place all these amino acid sequences which are being present over here. And normally you choose a range of interest in the sequence and select the database from pull down menu. Then keep the CD search checkbox on and leave options unchanged. That is, you use a default option and you will be able to find out what are the options of the NCBI BLAST. Now this is, you can just have a look about it, what you want.